Good.
Yeah.
All right, so it is my absolute pleasure to host Dr. Mark Hoddle for our seminar today. Mark was actually my master's advisor at UC Riverside, and now I get to list off some of his accolades before we get started.
Nothing embarrassing.
We got his bachelor's and master's of science in zoology at the University of Auckland in New Zealand. Did his PhD in entomology at UMass Amherst with Roy Van Driesch. Some of you might recognize that name. Recently in 2018, got his Doctor of Science from the University of Auckland. Other than degrees, he's been at Riverside since '97. I didn't realize it had been that long.
Yeah, it's a long time.
Doing extension specialist work, mostly in biocontrol and insect pests. He's worked on prominent pests, including glassy-winged sharpshooter, avocado seed moth, red palm weevil, and Asian citrus psyllid, which we'll be hearing about today. He's gotten many awards, laundry list of them, nothing to be embarrassed about. A lot for research and extension. Recently, he was named Fellow of the Entomological Society of America in 2018. That's still recent to me. And received an award for Distinguished Service and Outstanding Research from the UC Ag and Natural Resources Service just this year. So I'm all done with my listing.
Alison.
So
all right,
please.
Thank you. So thank you very much for the invitation. It is a real honor to be here at Purdue. This has been, you have to visit several, high-class entomology programs in the US. Definitely one. So it's great to be here at last. So I appreciate the invitation, and I thank you for coming to, giving me the opportunity to share the work that we've been doing in California with you. So the title of the presentation this morning is Crushing the Curve. This has come from the COVID stuff that we've been dealing with, because I was thinking about this curve crushing stuff in terms of population dynamics, and it's really applicable to applied entomology. We are always trying to suppress population curves, especially those of the pests, to levels that aren't going to cause us problems. What I want to talk to you today about is the work that we've been doing on this insect, the Asian citrus psyllid, which vectors a pathogen which causes a lethal disease in citrus in California. So this is the star of the show. This is Diaphragma citri, the Asian citrus psyllid. It's not native to California, obviously. It's probably native to the Indian subcontinent, and there's some controversy about as to exactly where the true area of origin is. But globally, I think most people have agreed it probably is the Indian subcontinent. So that's through Afghanistan, Pakistan, through India, maybe right down to the southern parts of Indonesia. I'm going to talk to you a bit today about the work that we did in Pakistan, looking for natural enemies of this parasitoid. And this is Tamarixia radiata, which has proved to be an extremely effective parasitoid of the nymphs of Asian citrus psyllid. And the Biocontrol program has suffered some disruption because of an invasive ant that's been present in California since around 1908. This is the Argentine ant, again, not native to California, but imported into the state. First showed up about 20 miles from Riverside, where I'm based in Southern California in Ontario, probably in shipments of soil that was moved transcontinentally in railway carts. And we'll finish up with some predator work that we've been doing on improving the control of this pest through the provisionment of floral resources in these orchards. This is the hoverfly larva, which is eating an nascent pea nymph. So if you're not familiar with the life cycle of psyllids, they're actually pretty simple. They're phloem feeding insects. The females can only lay their eggs in flush growth, which is the very tender growth that citrus produces, about twice per year. And then once those eggs hatch, there are 5 nymphal instars. And it's during this stage that the Asian citrus psyllid can acquire the bacterium Candidatus liberibacter asiaticus, which causes the lethal citrus disease Huang Long Bing. Once these nymphs are infected, they're infected for life, and they tend to be much more efficient vectors of the pathogen in comparison to an adult, which is uninfected but acquires the bacterium later in life as an adult. And then these adults can live in the orchard for several months as long as there are young green stems for them to feed on. But in the absence of citrus flush, there's no reproduction by these insects. They need that young tender foliage on which to lay their eggs. This is the bacterium. It's a phloem-dwelling bacterium, and once it's in those phloem conducting tubes, it tends to clog them up, and this leads to a variety of physiological disorders. The common name for the disease is citrus greening. We don't use that term in California. We prefer to use Huang Long Bing. There was a very interesting social science study done on this, when the pathogen was first detected, the Citrus Research Board gathered together a bunch of people in a room like this. So we've got this terrible disease problem. We told you your citrus had citrus greening. Would that be of a concern to you? A lot of people said, no, that actually sounds pretty good. Don't we want our citrus trees to be green? So then they said, well, how about if we told you the disease was called Huang Long B? Would that cause you some concern? It's like, can't even pronounce that name. That means nothing to me. Then they said, well, how about we call it HLB? Oh, that sounds kind of like HIV. Yeah, that sounds kind of like deadly disease for the citrus trees in California. So we refer to the disease as HLB and not citrus greening because of the negative connotations of a disease-causing organism. These will become regularly modeled with this type of patterning, and then within about 5 to 8 years, the trees die. There's no cure for this. Once your tree's infected, it's a death sentence for the citrus trees. So Florida's had this problem for quite a while now, and it's a multi-billion-dollar industry in Florida worth about $9.3 billion. The insect was first found in 1998. Nothing was done to control the spread of this insect in Florida, because at the time, the pathogen wasn't known to be in the state. 7 years later, it showed up, and the disease escalated through Florida, and the economic consequences of that infection have been terrible. More than 10% of these citrus acreage has died because of the disease. Economic losses have been massive, more than $330 million a year have been estimated in losses, and numerous jobs, more than 8,000 jobs have been lost as the industry has contracted. A lot of people now are speculating that Florida may not have a viable long-term citrus industry anymore. So it's thought to be potentially on the verge of economic extinction in Florida. So Seaman and Rogers did a very interesting study in a social science study. It was like, why has Florida done such a bad job at managing ACP and HLB when a country like Brazil, which has very similar growing conditions, climate, has done so well at managing the disease in the insect? And the decision, well, I guess the conclusion they reached from their work was that it was just a lackadaisical attitude amongst industry members to controlling the insect and the disease. And they put it down to a poor level of collective action for the public good. So some people are doing something, a lot of people are doing nothing. And as a consequence of those doing nothing, the industry has gone into this catastrophic downward spiral. California has watched all of this. We have a big citrus industry in California. It's worth about $3.3 billion. That's just the raw value of the fruit. But if you look at all the associated economic activity associated with that, it's about a $7 billion a year industry for California. Psyllid showed up in 2008, San Diego County, very southern end of the state, near the Mexico border. 4 years later, HLB was detected in Los Angeles County. The reason that the disease had shown up is that people had smuggled infected plants from China. There was a horticultural club associated with a church, and they'd been sharing infected material which they had been grafting onto trees. So we ended up with a widespread infection. A lot of those trees could be tracked, and they were roved out and destroyed. So what's been the impact in California so far on our citrus industry? And that's going to be the basis of the discussion this morning. We've spent tens of millions of dollars, probably up to hundreds of millions of dollars now mitigating this issue. And HLB, the disease, and the insects so far are almost exclusively a problem of urban citrus. Just about every house in California has 1 or 2 citrus trees either growing in the backyard, or on the side of the house, or in many areas they're grown as ornamental trees as well, around parks and shopping malls and this type of thing. So a massive reservoir. So why have we been successful at maintaining it in these urban areas? There's been no massive outbreaks in commercial citrus. And I'm going to get to some of the reasons as to why I think that has occurred. We're going to start our story here in Pakistan, where we began the foreign exploration efforts for Asian citrus psyllid. Our base of operations was here in Faisalabad. The University of Agriculture is based there. There's a big entomology department. Unfortunately for us, the chancellor of that university was a Riverside graduate who worked on Huang Long B and other citrus-related diseases, both in California and in Florida. So basically, the doors of that institution were thrown open to us when we said we were coming there to work. Our field sites were around Faisalabad, so we'd fly into Lahore, drive a couple of hours to Faisalabad. Then these major citrus growing areas are here in Sedota. Gujranwala, and down here in Toba Texas. And this is where we were looking for parasitoids of Asian citrus fruits, part of the native range, the Indian subcontinent, which stretches right through this area and down into this part of Southeast Asia. So when we got there, when you go out to these citrus orchards, I was expecting to see wastelands of citrus. It's not the case. In the area where the disease and the insect are endemic, citrus still flourishes. And the big question is, why has citrus flourished in these areas, whereas it has gone into such a strong decline in Florida? But that's a whole other story, which we won't have time to get into today. Working in Pakistan would be a little dodgy at times. There's some areas we would go into, we were given these elite commando units. We'd walk around protecting us, but really unnecessary. People were very friendly, and we had no troubles while we were working there. If you're not familiar with parasitoids of Psyllid nymphs, they're extremely small. And this is what we were looking for in the field. People go, how on earth do you find these tiny little insects? If you know how to do it and what you're looking for, it's actually pretty simple. So if you're not familiar with the life cycle, I'll quickly detail this for you now. Our star of the story, Panorexia radiata, is an ectoparasitoid. So females lay their eggs underneath the nymph. Maybe you can see one of the eggs there attached behind cocci with that Psyllid nymph. We've flipped it over so you can see it. The grub hatches out, begins feeding on the belly of the nymph, excavates its host, basically eviscerates it, completes its feeding inside the host. It then pupates within the eviscerated host. This is the pupa. You can see we've flipped it over. And if you're looking at the dorsal side, you'd see that the mummy is this brown color, and then the parasitoid spins these silken strands around the outside of the host to hold the shell over the pupa so it can pupate within a protected environment. When they get ready to emerge, they chew these circular holes. Panorexia, parasitoid we brought back, which established readily, emerges from the anterior end. The parasitoid Allison worked on, unfortunately, did not establish. We still learned a lot about it. Emerges from the posterior end of the host. So when we're in the field, especially in Pakistan, looking for these parasitoids, we could readily identify who was doing the killing based on the position of those emergence holes. These parasitoids can kill in a second way too. This is called host feeding. In this instance, the female uses her ovipositor like a dagger. She'll stand on the back of the host, pierce the dorsum of the host multiple times until the hemolymph starts welling out of it, and then she'll turn and feed on that insect blood, and this provides the protein that she needs to develop more eggs. So they kill in two ways, through this process that we call parasitism, and then through a secondary process where the parasitoid acts more like a predator and kills it through feeding. Now, one thing that we did when we went to Florida, we reviewed the genetics literature on how do you preserve genetic diversity in natural enemies. And a paper was published by Rick Rausch and Keith Hopper way back in 1995 gave us some ideas on how to do this. When I went to Pakistan, each time I came back with natural enemies, those collections represented a different snapshot in time, both temporally and spatially. And each of those collections was set up in its own cage. So we had 17 of those cages in the quarantine facility, and they were bred back in on themselves. So we were encouraging inbreeding with the idea that each of these lines would become homozygous. So a certain amount of genetic diversity would be lost. Then once they were homozygous, there's no more genetic diversity in those cages. Those alleles would become fixed, and those parasitoids could not become adapted to the prevailing conditions in quarantine. This has been a criticism of a lot of biocontrol programs. Get your parasites from all over the place, you put them in a box, you let them breed up, they keep swapping genetic material, then they adapt to the quarantine facilities, you go to put them outside and they don't do a very good job because they have just become domesticated to the cushy conditions that they have in the quarantine facility. So to reconstitute that randomly preserved genetic material, we would take individuals from each of those inbred cages, put them into what we call the mongrel cage or a panmictic breeding cage, and just allow random matings, which would reconstitute whatever genetic variation had been captured in these cages. Then those hybrids were released into the field, and then natural selection would act on those hybrids. And the idea would be that the prevailing environmental conditions would select the most effective, strongest parasitoids. So whether we were releasing them in cool coastal areas, the warmer interior areas, or in the friggin' hot areas out in the desert, there should have been enough genetic diversity to allow those parasitoids to proliferate. One thing I didn't tell you about was Pakistan was also chosen because it has about a 70% climate match with the major citrus producing areas in Pakistan. And you want your natural enemies to be pre-adapted to those conditions that they're going to be released into. Okay, we did all the quarantine stuff, got the permission to release after all the host safety testing was done, and the process was very simple. We mass reared them. We went out into urban areas, say like here in Los Angeles or around Riverside County, tied up the vials of the parasitoids and the trees, opened the lids, just let them out. There were so many psyllids around. They didn't have to move very far to find food. So our first releases began in the middle of winter in December of 2011. This parasitoid is extremely sticky. It established at those release sites immediately. We came back 2-3 weeks later. I couldn't believe it. There were exit holes everywhere in those nymphs. This is amazing. And as we moved into new areas to release the parasitoid, it was already there, 5 to 8 miles away from some of our nearest release sites. So then the question was, well, you're finding all that stuff in the field. Is it your parasitoid from Pakistan? The psyllid came in on its own. Did a parasitoid hitchhike in with that psyllid from somewhere else? So we did the genetics work, and everything we were recovering in the field had a very strong genetic signature from Pakistan. It's the only place in the world you can find it outside of its native range of Southern California. Florida, all these other places you can see from the haplotype network tree here, don't come close to the Pakistan types that we were releasing. So what we had put out in the field was our parasitoid. It had established on its own and it was spread. So far, we've released more than 23 million of these parasitoids. Now this project is being run by the California Department of Food and Ag. Once we got the initial releases done in the establishment, we couldn't rear enough of them, so we just turned that over to the California Department of Food and Ag. Well, the big question is, okay, well, that's great. Put your parasites out, they're from Pakistan, you found, well, are they doing anything? Impacts. So we did 3 things. We would go out every month and we would count psyllids and look for parasites. We ran some protected exposure studies, some basic life table stuff. And then we ran what I thought was some very cool videography studies that we did with these little Raspberry Pi cameras that we set up in the field, where we would video 24 hours a day for several weeks, those ACP cohorts, to see who was coming to them and doing the killing in those life table studies. So let's go through those data now. Virtually all the sites that we worked at have a data set that looks like this. Prior to the Biocontrol program, lots and hundreds of psyllids in those citrus trees. Nothing was controlling them, no evidence of parasitism at all. Parasitoid is released and establishes, and you can see a very rapid decline in psyllid numbers, and they never recover. And each time there's a peak in psyllid population dynamics, here the parasitoid responds quite strongly. We've run 2 separate studies, 3 to 4 years each, over multiple sites throughout Southern California, and our data suggests that following the introduction of this parasitoid, ACP populations have declined by at least 70%, almost certainly due to natural enemy activity. Okay, so another bigger study that we ran, 28 sites, 55 trees. The work that went into this has been intensive, and it's just not captured in this single slide. Egg densities have declined by 92%. Small nymphs have gone down by 81%. Large nymphs, the population densities over this period declined by 94%. Adult densities declined by 75%. Parasitism, around 20 to 25%. During the peaks of ACP activity, parasitism would jump to over 60%. These are extremely good results for us. However, when the Argentine ant is present, we notice that it has an extremely strong and detrimental effect on the efficacy of these biocontrol agents. Ants are present, eggs, that should be eggs, small nymphs, big nymphs are about 3 times bigger. And the reason for that is those nymphs are producing honeydew, which is a sugary waste product. Ants love harvesting that. And in return for that food, they will protect their micro livestock. They'll protect their micro livestock from those natural enemies. No association with adults, because adult nymphs don't produce a sugary product that the ants can readily harvest. So we think that the impacts of the parasitoid may be underestimated because we can't measure in the field impacts from host feeding, the predation effects those parasitoids have. You can count exit holes easily, but if you find a shriveled up nymph on a tree, you don't know what killed it. Was it host feeding by the parasitoid or did something else kill it? That's where the videography comes in. The other thing I loved about this study, if you're into a biocontrol and density-dependent mortality, we're looking for delayed density-dependent effects. So our work suggests that what the parasitoid was doing in year 0, the population level impacts would not be manifested until 12 months later. So you'd see a delay of about 12 months before the population level impact was observed the following year. I love that result from the study. Okay, so let's do some finer resolution here through these life table studies. We would put out small potted plants infested with Asian citrus psyllid nymphs that go into the orchards. We'd have a control plant, which had a bag around it, which stopped all sorts of natural enemies getting in, so the ACP could develop in the absence of natural enemies. We had plants with tanglefoot around the base, so only flying natural enemies could get there, like Tamarixia, maybe hoverflies as well. And we had this exclusion cage, which had a mesh that was big enough to allow the parasitoids in, but keep out all the bigger types of predators, say like lacewings and surfeit adults, for example. Then we had the final treatment, which was a free-for-all. Anybody who could find that free food was welcome to come and feed on it. Erica Kistner, a postdoc in my lab, did a tremendous job on this. We ran these studies, spring, summer, fall, and winter. 103 of these cohorts were deployed. We did 3 sites. And I'm going to show you one of these survivorship curves, which is just from the summer data, but the data are all very similar. In the absence of natural enemies, about 90% of the ACP survive. If Tamarixia only can get to those nymphs, it'll kill about 50% of them. If all the other types of natural enemies that can fly and walk can get to those ACP cohorts, they'll kill around 70 to 80% of those ACP. So natural enemies have a very strong regulatory effect on populations of this pest in California citrus orchards. But it didn't answer the questions when we were finding dead nymphs on those trees that we put out. What killed them when there was a small withered up brown body on that branch? So a tech in my lab, Mike Lewis, made these little micro cameras, which we would focus on the ACP cohorts that we were putting out there. The Raspberry Pi technology is very cheap to buy and it's pretty robust in the field. Cameras could record for about 5 days. We'd swap out the thumb drive, replace a new one. We had infrared LEDs put around the lens so we could film during the day and the night. And the resolution is pretty high, around 5 megapixels, so pretty decent for looking at. Okay, so what did we find out? We had 19,200 hours of video data to look at. We had a crew for about 6 months. This was a full-time job. 2 screens sitting in the lab, fast forwarding through all this video, looking for those rare events that would happen not very often. And as I was leaving for the day, put my head in the lab and go, hey, you guys see any, was it Higgs boson? You know, these ultra rare events in physics, right? It's like looking for one of these. We did record 647 unambiguous kill events. Much to our surprise, about 60% of the killing was done by these surfeit fly larvae, which we never saw because they're nocturnal. We were never out in the orchards at night. We had no idea they were out there doing this stuff. About a third of the kills were from our parasitoid, and then about 12% of the kills were coming from lacewing larvae. No evidence of spiders doing anything in the system at all. Although you read the literature, spiders are thought to be important generalist predators of ACP. We have no evidence for that. And even in the home range in Pakistan, where we ran a similar set of studies for 2 years, we saw no strong association with spiders having any impacts on this pest at all. So that's a myth that needs to be dispelled from the ACP literature. 60% of these predation events were occurring at night. Our parasitoid, interestingly, was most active between 10 a.m. and 1 o'clock in the afternoon, about 3 hours, and you don't see it anymore. So that was another interesting result from the study. But the videography also revealed something we've been observing in the field, was that Argentine ants are almost always present on those ACP colonies. If they can get to them, they will take those dry columns of honeydew and take it back to the nest. So here are the ants, and this is what they were looking for, this honeydew. Now, these psyllid nymphs, unlike a lot of other honeydew-producing insects, produce their honeydew as a dried ribbon. So they've got to clip it and carry it back to the nest in their mandibles. This ant is a specialized liquid sugar feeder. It loves taking liquid sugars from, say, ground soft scales and mealybugs, drinking it, and then carrying it in its gaster back to the nest. Very efficient, like railway carriages moving along a line. But this is a slow process. But since there's so many ants in the field and they're always so hungry, they'll drop down to a less preferred honeydew source. And here they are, tending them. They form this disruptive mutualism with these nymphs. They'll protect them from natural enemies, and in return, they get rewarded with the honeydew. About 90% of our orchards are infested with this. We've done some estimates where we suggest that suggest to us that some of these trees may have over 1 million ant visits a day. Astonishing levels of protection in these trees from this invasive ant. About 90% of these sap-feeding pest colonies like mealybugs, soft scales, aphids, for example, are protected, and about 55% of Asian citrus psyllid colonies are protected, mainly because that reduction is because it's a dried honeydew source and not a nice liquid one that they can easily drink. So some of the benefits from this: removing the honeydew increases survivorship of the pests because they're not going to be subjected to fungal infections, so sanitation is important. Interestingly, there's evidence to suggest that when these ants are harvesting the honeydew, they increase the development and reproduction outputs of these livestock. Because if they don't produce enough, they get killed and eaten. So there's a lot of pressure on them to feed more, give them more honeydew if you want to survive. And then these ants, once the colonies get too big, they'll start moving them to new parts of the orchards and starting new colonies. So now the ant colonies are growing bigger because they're getting lots of food, and their farming enterprises are expanding throughout the orchards as well. And for us, the big problem is natural enemy interference. And here you can see in this video, the Argentine ants patrolling and attacking and killing our parasitoid. This is not what you want to see when those parasitoids is worth over $1 each. And the ants are you're basically feeding the ants protein that they take back to feed the brood in those subterranean nests. Okay. How many ants running up and down a tree trunk is going to be a problem? Kelsey Shull, a PhD student in my lab, I'm going to see some of her data next, did an extremely good job at this. Her estimates suggest that maybe just 10 to 20 ants a minute running up and down a tree trunk are too many ants. We now have a much larger data set, which is suggesting that maybe 50 to 60 ants a minute running up and down the tree trunk is probably where the action threshold for ant control really lies. So the question Kelsey had was, if we go out to the orchards and we control the ants using these liquid baits, can we improve the levels of biocontrol and see these pest populations drop? Amazingly, this hasn't been done very effectively in citrus orchards before. So we would use these bait stations, which we would fill with a 25% sucrose solution and a tiny amount of insecticide, 0.0001% thiamethoxam. The ants drink from this, take it back to the nest, feed these sublethal doses to their workers and other nestmates. The toxins build up over time, and then you get these catastrophic colony collapses throughout the orchard. So 2-3 days later, the ants have gone. So Kelsey ran these big experiments where we had commercial plots. She'd treat some of these trees with ant bait stations and others were left as controls. 18 plots, 108 trees examined every month for the presence of ants, presence of sap-sucking pests, and any natural enemy activity in those orchards. So let's have a look at what we did. Every month, we'd go out and take our samples from the trees. We looked at 3 different sites: flush, gun-green stems and twigs, and fruit that were infested by 4 different groups of pests. And we'll get to those in a minute. We'd count the number of ants running up and down the tree trunks for a 1-minute period in those treated and untreated blocks. And then in the lab with the samples that we collected, we'd look for evidence of natural enemies, record the ACP stages, and then we'd look at the citrus mealybug, Asian citrus psyllid, brown soft scale, and California red scale. This is not a honeydew producer. Hard scales of the diaspids don't produce honeydew, but they benefit from protection from ants because ants, as they're patrolling those infested areas, this pest co-inhabits some of those substrates where there are honeydew producing insects. They benefit indirectly from these patrolling ants and they scare away of Phytis malinus, which you may know about as a very important natural enemy of these California red scales in California. Okay, so what did we see with the baiting program? Statistically equivalent levels of pests, ants, in those control and treatment plots before the sugar order was put out. 1 month later, a 66% reduction. 2 months later, ant numbers have been reduced by 92%, 20-fold reduction in ant densities, and we maintained those ants below that action threshold for the entirety of the study, 18 months in all. So controlling the ants didn't do anything to those pest densities. So in untreated trees, there was no difference in pest densities between the twigs, flush, and fruit that we were monitoring. Afterwards, a 97% reduction in pest densities on the twigs, 84% in the flush, and 99% on fruit. Just taking out the ants allowed those natural enemies to do an amazing job at controlling those pests. The biocontrol program that had been developed for these pests in citrus in the 1920s, 30s, 40s, and 50s, and even into the 60s, is extremely effective. Those old guys like Paul DeBark and the rest of them, they found great natural enemies. They never realized their full potential because Argentine ant had always been in that system. Okay, so look at the pest densities, and you can see that all of them, huge reductions in numbers between treated and untreated trees, except for our key pest. Asian citrus psyllid densities didn't change in the presence or the absence of ants. This was kind of a crushing result for us. But Kelsey had an interesting way of looking at this, and I'll get to that next. So even though this result was sort of disappointing, colony size didn't change. As what she did show is that the percentage of flush growth that was infested changed a lot. So even if colony size of ACP did not change in the absence of ants, the percentage of flush growth that was being attacked by those ACP nymphs declined by 75%. And amazingly, with citrus mealybug, we entirely eradicated that pest from those study sites. The irony of this study, from a biocontrol point of view, was we were seeing far higher levels of parasitism in the ant plots than in the treated plots, because in the treated plots, there was nothing for the natural enemies to attack. They'd cleaned out all their prey and hosts. So we had to get our parasitism estimates from the control plots where there was still pests for those natural enemies to attack because the ants had protected those hosts. Okay, so what happened? Why did we see this? We think parasitism and natural enemy presence was a main driver for this decline. 6 weeks after killing off those ants, we saw a huge 9-fold increase in natural enemy activity in those ant-treated plots. And then those natural enemies disappeared. The reason they disappeared was there was no food for them to keep them around any longer. And the pest populations never recovered in those treated plots for the duration of that study, and those bait stations were being replaced every month. We had wicked ant densities in these orchards. So reinvasion pressure from untreated areas was enormous, so we had to keep up the baiting program. But this was to be done in the commercial orchard at a full-scale level. It would be an area-wide control program. And you probably wouldn't have to bait for the ants as intensively once you clean them out through the entire orchard. So let's get to this guy, the syrphid flies. We'd seen from Erica's work with videography that syrphid fly larvae were important. They come out at night, you don't see them. Well, when we were sampling the flush, we were finding syrphids in the flush. The larvae were hidden down in there. And they cycled very strongly with ACP. So you can see here in the green, the target pest, and here in the orange, what the psyllid numbers were doing. So pretty strong evidence, I think, to suggest that syrphid fly populations cycle with the availability of that Asian citrus psyllid prey. These liquid bait stations suck for controlling ants. They are expensive to buy, $20 each. They have extremely high maintenance costs. You need to collect them every month. You need to clean them. You need to make new bait, put the bait back in there, take them back to the field, and redeploy them again. The bait ferments when it's hot. There being reports of these canisters blowing up when it gets really hot because the gases are just trapped in there. That didn't happen to us. As it evaporates, the sugar concentration changes. These ants are very fussy. They will no longer feed from them. And as it gets more concentrated and sticky, as the dead ants start piling up around the margins of the trap, the station, they won't go in there anymore. So you're constantly cleaning these things. So the question then became, can we design a better ant bait delivery system? So I talked to some engineers in the chemical engineering department about this. Oh, of course, this is simple. We do this stuff all the time. So, really? They said, yes. And I said, well, okay, can we invent a biodegradable bait station? We just throw onto trees like slug and snail pellets. Oh, of course, we can do that. All right, let's do it then. So we developed these biodegradable hydrogel beads using seaweed extracts. And they hold the toxic liquid with the insecticide, and the ants recruit to them when we throw them underneath the trees. And they feed themselves. In about 2 to 3 days, these gels dissolve into the soil and they're gone. Here's a close-up of the ant taking the liquid out of those gels. It's the same solution that we were putting in the bait station. We can infuse those gels with that poisoned sugar water. Very simple to make in the lab. Products are relatively cheap. Make the beads, put them in the liquid with the sugar water and the insecticide. 24 hours later, they've fully absorbed that liquid, package them up, take them out to the field, put them underneath the trees, and the ants recruit to them immediately. So you can see these are the densities of ants that we're dealing with in those orchards. And within 2 to 3 days, the beads are gone and the ants are not doing very well. So Kelsey ran another field study looking at similar sorts of things. When you put the beads out over a 3-month period, with beads being put out 3 times, three weeks apart, you can reduce ant densities from those crazy high numbers by about 91%. And you can see there's some persistence there after the last application. Didn't quite get below our action threshold, but I think a great proof of concept for the first time this has ever been done in a citrus orchard. Okay, so a question I get asked a lot is, will those gels attract other sugar-feeding insects like bees? I don't think they can, and we've seen no evidence of that in the field, because what's going to feed on those gels when they're covered with that many ants, and they're very aggressive towards anything that wants to take their sugar food? So we've seen no target impacts. We're going to set up those micro video cameras over these gels next spring to document that more effectively. Okay, we can kill the ants. We've sort of got an idea of an action threshold. If anybody's done integrated pest management, you know you're going to have to go out and count stuff if you're going to implement some sort of management program. Do I have any volunteers here to sit underneath these trees to count ants? A horrible job. Extremely boring. The ants are dripping out of the trees, landing all over. You stand up. Some of these citrus varieties, like these grapefruits, can have big thorns on them. They rip your clothes and they pierce the skin on your backs, or if you're not paying attention to where you're walking, they'll go through the soles of your shoes. So here's Ivana and Francesca, and we're sitting underneath the trees counting ants. Ants love running along these irrigation pipes. They get to the tree trunk, they go up, collect their honeydew, come back down, run along the pipe again, back to their subterranean nest. This is very stereotypical behavior, and we're going to exploit that in the next set of slides. These are the densities of ants you'll see on those irrigation lines. Studies by NASA space engineers, I don't know why they did this, looked at this phenomenon. And I think they might have been building robots to walk on Mars. So they knew that these ants were very good at doing a couple of things. Finding a linear line to get to their food, because that maximizes foraging efficiency, and the ants hate running over uneven ground. So putting out these long, hundreds and hundreds of yards of straight irrigation pipe that's smooth gives them a super highway to run back and forth across as fast as they can. So these NASA guys 3D printed these little plates and they make the ants walk over them. And what they found was that as the terrain became more and more uneven, it would slow their walking speeds or running speeds by up to 42%. So that suggests strongly the reasons why the ants like using those irrigation pipes. They're straight and they're smooth. We're going to come back to that uneven terrain issue at the end of this presentation. As I said, nobody wants to sit underneath these trees counting ants. It's a horrible job, but we know the ants are always on those pipes running back and forwards. So after talking to the chemical engineers, I went down to the computer science department and said, hey, we know these ants are always on the pipes. Do you think we can invent something we could put on the pipe that could count the ants for us? Oh, of course we can. That's easy. Actually, we'll get an honors student to do that. Thanks. So what we ended up doing was we 3D printed these clamps, put them on the pipe, And in this box was some computer software. And then in this area, we had these infrared beams that would span across that pipe. And each time the ants run through it, they break the beams. So it's like walking into a shop, you hear the bell chime and alerts the attendant somebody's come in. Exactly what's going on here. All those data, let me show you this. I pulled one apart and hooked up this little light to it. So each time it's flashing, an ant's being counted as it breaks that infrared beam. So every hour on the hour, for 1 minute, those sensors turn themselves on. They count the number of ants that are running through the pipes. Then all those data at the end of the day are transmitted to this thing that's called a gateway. So the sensors talk to the gateway, data comes in, and then all that data is sent to the cloud. And we have an app, an ant counting app that you can look at on your phone. And it can tell you what the levels of ant activity are in different sectors of the orchard. If you cross that action threshold, you go out and treat the ants. You don't have to go and count anything. You can sit in your air-conditioned office, tracking in near real time what the ants are doing in different parts of the orchard. And these are what the data look like. Here's some count data. You can see for the most part, some of those areas are well below the action threshold, some are crossed. We can outfit those sensors with humidity and temperature readers so we can get very precise environmental data on what's going on in different parts of the orchard that may affect your irrigation plans, for example. We can generate heat maps, see how ant activity in those different areas is changing through the course of the day. How do the environmental conditions, for example, affect those ants? How effective are your treatments after you put them out? How quickly do you drive ant numbers down to low levels? Do you need to reapply quickly, or can you delay it for a while? So you can do all that now from your office. That's how I see this whole thing working in the future. It should be fully automated. The sensors talk to a machine in a packing house. That machine, like some sort of fancy cappuccino machine, makes those beads for you. Those beads drop into the back of some type of land drone. That land drone has the GPS coordinates and goes out to those precise trees and dumps those gels underneath the trees. It can all be automated. You do it at night when temperatures are low, humidity is high, that preserves or increases the longevity of those bees, and there's no people in the orchard, so you get around all your re-entry interval issues through treating for ants. Okay, so let's get to this crushing the curve part. We're almost done here. No pest control. You cross the economic injury level, and no actions taken to suppress those pest populations once the action threshold is suppressed. Just say if we mitigate some of those problems by controlling the ants, maybe we still cross the action threshold, but we don't reach economic injury. Can we synergize this and make the system even more effective? And that's what we're working on now. Could we enhance biocontrol so your pest levels are never crossing the action threshold at all? Can we get more value out of the natural enemies that are naturally occurring in those orchards? And that's what we're doing now. This is what our orchards look like, barren. There's nothing in there, very hostile environments. A lot of work out of the conservation biocontrol community suggests strongly that if you put some sort of resource in here, like a simple thing, like some flowering plants that provide nectar and pollen, natural enemies will respond very strongly to those resources and their populations will increase and you'll get improved levels of pest control. So Nick Irvin in the lab has been working on evaluating different types of flowers that are adapted to the Mediterranean climate that we grow our crop in and that are reasonably drought tolerant. So we've looked at native California poppies, Psyllid, alyssum, and buckwheat. And she's out there counting hoverflies that are being attracted to those flowers. This is Ag Ops near UC Riverside, about a mile away. Hostile, hot, barren environment. These flowers are planted literally in the middle of nowhere. But those flies, those hoverflies, those surfers, find those flowers. We've got no idea where they're coming from, but they respond strongly to these resources when they're put out in the field. So what we found was the California natives, California poppy and Psyllid don't attract a lot of hoverflies. Buckwheat does a reasonable job, but this one, a Mediterranean specialist alyssum, attracts a lot of hoverflies. So a very good resource for those predators. So Nick ran some studies in these citrus orchards where, again, we put out our potted plants with ACP on them. And she put them next to plots that had alyssum and these control plots that lacked those flowering resources. She'd go out, count the numbers of surviving nymphs on those plants, count the number of hoverfly eggs and larvae on those plants. We even saw hoverfly larvae coming into those plots and ovipositing out in the field. Amazingly, here is a first instar hoverfly larva eating a fifth instar ACP nymph. So a little guy is eating an enormous guy there, which is pretty cool. Okay, so what effect did it have? When the flowers are present, ACP survivorship on those little plants goes down significantly. You'll see 3.5 times more hoverfly eggs on those ACP patches. Hoverfly larvae will increase 2.5-fold, and ACP survivorship declines significantly. So great. So what conclusions can we take from this? I asked at the beginning of this topic, of this presentation, the question is, have we crushed the ACP curve in California? I think we have. The biocontrol agents have played a major role in flattening out those ACP populations. Those urban areas where we established that natural enemy quickly, they were massive reservoirs for the ACP and the disease. And we were very worried about billions of ACP blowing out of those areas and going into those commercial orchards. You can't go through and spray hundreds of square miles of highly urbanized citrus for these pests. And that's a whole other story about homeowner resistance to those programs. Everybody liked the idea of the biocontrol agents going in, and nobody would ever see them doing their job. So we've seen at least a 70% reduction in ACP numbers after the biocontrol program began. We've demonstrated in orchards where we have seen some ACP activity, but not massive infestation, not huge swaths of California infested, but these ants, when you control them, release enormous biocontrol potential to the point where you may not even have to spray in many instances because the natural enemies effectively eradicate a lot of those key commiferan pests. And we've had no major ACP or HL3 outbreaks in commercial California citrus, largely because I think the populations in those urban areas were driven down to such low levels and have been maintained there. Now, hoverflies are very important predators, and we can enhance their efficacy in those barren citrus orchards by putting out very small plots of these flowering plants. So what are we doing now? Right now, we have gone all in on these sensors. We've got big grants through the Department of Pesticide Regulations in California. DPR has banned the use of chlorpyrifos, which is a broad spectrum contact insecticide in citrus. It was the only registered product for ant control in citrus orchards, so there is nothing now. Their idea is to drive innovation through legislation. That's what it's called. So you take away chlorpyrifos, we'll put up millions of dollars of grant money. We want you clever guys at the universities and other places to figure out alternative ways to control those ants if we take chlorpyrifos away from them. So at least they put their money where their mouth is. So we've done these large-scale sensor deployments, and we're demonstrating the use of those to growers. We're now doing huge trials with these hydrogel beads through replicated orchards. We have big plantings of the alyssum throughout these orchards, and we're monitoring all those pests like I showed you in those smaller studies. I'll just finish up with, remember how the ants love running on those pipes? They're always there. You could really go after targeted ant control by focusing on the pipes. So our work now is looking at taking information from urban entomologists who do a lot of work on controlling pest ants in buildings. And they have a ton of literature on insecticide barriers. You put out a very low concentration of insecticide, ants pick up those molecules on their bodies, then horizontally transfer them back to the nest. You can do that on pipes. You don't have to spray the whole orchard, you can just spray a pipe. They'll carry that stuff back to the nest. We've identified about 4 or 5 compounds that work very effectively. The other thing we did, and we're in this right now, this is really cool. That work that was done by those NASA guys who looked at ants walking on those uneven plates that they were 3D printing, we can 3D print pipes with those same patterns, put them out in the orchard. We may not have to kill the ants. If we can just slow them down by 42%, the pressure that they're putting up in the trees will lessen. Then the final thing that we're looking at, just cover the pipes with organic mulch. This is good orchard practice, promotes soil health, retain water, and suppress weeds. So a lot of our efforts now are focusing on exploiting that stereotypical behavior of those ants, using those long runs of irrigation lines throughout the orchard. So we're trying to drive a solution here that reduces our use of insecticide, uses biocontrol, cultural management, and low rates of insecticides to control pest ants, promote biocontrol of things that are in the tree. So our whole philosophy now is controlling these pests from the ground up. These ants are on the ground, they live underground, we can control them on the ground, things up in the tree take care of themselves. I have a lot of people to thank, but given what's happening in Pakistan right now, a special call out to our friends and colleagues at the University of Agriculture in Faisalabad, for terrible flooding through there because of the heavy monsoon rains. Thankfully, that part of Punjab has not been adversely affected by the droughts, but now they're going to be looking at significant food shortages and catastrophic crop loss. Any questions? I'll be happy to take those for you. If you're interested in what's going on in these photos, it's a great story here and I'd love to tell that to you. Do we have any time? Am I done? Am I over time, Steve? No? Time?
No, I think we're scheduled here until 12:30, I think.
Oh, okay. Let's keep it. Okay, all right. Just want to tell you this story. You know, India and Pakistan are rivals, right? Nuclear rivals. And one of the reasons they have nuclear weapons is that Pakistan is very worried about India stealing its water from the Himalayan mountains that sit up in the north part of the country out of an area called Kashmir. It's where the 5 big rivers originate and flow down and irrigate Punjab and other highly fertile agricultural areas. So each day at the India-Pakistan border at a place called Wagawat, they have a ceremonial border closing. And these huge mammoth men on both sides of the border march up and down the roads doing these dramatic displays of military prowess, like big muscle flexing out there. And then when they're done, they line up and they slam those gates shut. You know, there's crowds on each side like cheering, whoa, going all crazy, waving their flags. You know, it's like a highly nationalistic event. So then afterwards, you can have your photos taken with some of these gargantuan soldiers. So I'll get my photo taken with that guy. So walk up and say, can they have your phone? Yes, sure. I'll let you shake your hand. Oh, okay. I'm shaking his hand. He's squeezing it hard. He's looking at me right in the eyes. Okay, I'll squeeze back too. So then he ramps up the pressure. Does it a third time. Like, see, my fingers are turning really white here. It's like this mammoth hand's like crushing my fingers. And he's staring me right in the eyes. And he says to me, good. Take the photo. That's when he turned and looked at the camera. That was just a classic experience about working in that place. It was just great, great, great time. So Christina, my wife, she came with me on all those trips. Really great field help, and we were there for 2.5, almost 3 years of working back and forth in Pakistan, up to a couple of months at a time, being there a few weeks, 2 months at a time. So a lot of stuff you hear about this country, just not true. And all the horror stories you hear on the news are rare events, but working day to day with the people, living with them, being out in the fields, camels, mules going back and forth, sitting under the trees, working on this, listening to people walking by, going about their lives. Amazing stuff. All right. If there's any questions, I'd be happy to answer them. Steve, yeah, easy question. This is not my, no oral exam.
Good questions, actually, too. One is, so have the growers done anything other than accept biocontrol at this point? In other words, have they made any modifications based on all of the things that you could be implementing at this point?
Great question. So this is now the extension phase of what we're doing.
Yeah.
We are having workshops now where we're going out to the field and we're outlaying all of this technology and explaining it to them.
Right.
The growers love these hydrogel beads.
Okay, because it's easy and cheap.
Very easy and cheap. The problem is now it's not regulated. We don't have permission to deploy the insecticides in this way. So now we're working with DPR, Department of Pesticide Regulations, to sort of like loosen up this. So each county has an agricultural commissioner who oversees insecticide use in their particular county. So the ag commissioners that we work with have just turned a blind eye to growers doing this stuff themselves. They've said, the insecticide rates that you're using is, look, that level of thiamethoxam that we deliberately put out on a per acre basis, is less than the minimal acceptable residue levels for an organically certified orchard. So if you're an organic orchard and you just naturally had that level of thiamethoxam and it was higher than what we were putting, you're still organically certified. Even if we go in there and put much lower rate, we can't do it because we are deliberately putting a neonate into those organic orchards, even though it's at rates that are much lower than you're allowed to have naturally occurring. The dust is blowing in from your neighbor's place. It's just mind blowing.
The other question I had is this interface between regulations and biology. You talked about the challenge we have in classical biocontrol when you bring in a predator or parasitoid, all the regulatory hurdles that you go through to get it into culture. And you don't have permission to release it right away. Some of these things sit in cultures and colonies for decades.
Yes,
Decades.
right.
So it's not a surprise that the inbreeding overwhelms a lot of this population. What I don't see in the field is evidence where we're monitoring some measure of viability as these populations are established and maintained over time. The way we overcame it when I was working on introduced natural enemies in Africa is we constantly introduced new genes by introducing new populations, and like you're doing with these mix between two populations, right? Did you ever make any measurements to understand, what sort of outcome or outputs you are hoping for and can continue to monitor?
Right. Great question. Right. I'm going to answer that in two parts. Nobody knows what the fitness genes are in these insects. So trying to monitor those through genetics work was going to be near impossible to figure out. So I submitted a grant, which wasn't funded, unfortunately, and it was called Let the Genes Do the Talking For Us. Whereas if we were putting out these hybrids and we're expecting to see them become adapted to cool coastal, moderate interior, and hot desert areas, if that whole natural selection process had worked through releasing those hybrids, we should be able to take representatives from those three different populations and subject them to different temperature regimens, mix and match them, and see whether or not they performed according to the expected hypotheses that we would have generated from genetic reconstitution and natural selection acting in the field. There's a key question that needs to be answered. And it's critical because, as you can imagine, running a program like that, incredibly expensive, very frustrating in terms of having enough supply of plants and susceptible psyllids to be moving into those 17 colonies all the time. And if you don't need to do it, well, we need as I've written on this stuff, this whole idea, intellectually very appealing. But we have no field evidence at all that it is worth doing. And we have not been able to get the grant money for it because they established it doing well. What do we need to do any follow-up work for? You're done. That's where we sit with that stuff.
Thank you.
Great question. Yes.
So with your hydrogels, the rate is exceptionally low for thiamethoxam. Like most ant gels are at least a factor, maybe two higher than that.
Yeah.
Why did you go with such a low use rate?
Right. So again, we just poached all that work from the urban entomologists who've done a ton of this type of work, looking at controlling Argentine ants around urban households. So we used their rates, and then we ran even more experiments looking at even reduced levels of rates. So with thiamethoxam and spinosad, for example, you can go to half of that and still get good kill. The downside is it takes several days longer. And because of that increased time, the gels are starting to shrivel up and losing their attractiveness to the ants. But if you were to keep it in a liquid bait station, those even lower rates would still be effective. So we're just finishing those field trials now with those much lower rates. And in the lab, it works great. But once they're out in the field and the gels are subjected to the field conditions where they desiccate quickly, and the ants, there's just so many ants feeding on this stuff. And there's a trade-off between feed longevity, insect concentration, and then time to kill. Because if that time to kill is too long, the gels have lost their effectiveness whatever insecticide is building up in the colony is now dissipating, and they've gone straight back to their honeydew producing hosts again. So that's really a critical, ratio of at least 3 or 4 things that you got to get right.
Yeah. Are you out anything by going a little higher with your rate if time's a factor and that time to kill?
That's a good question. We could possibly go a little bit higher, but I think what the urban entomologists have shown is that it's a very fine line between killing them too fast and not getting enough material back. See, the difficulty with Argentine ant, this is just another whole fascinating talk, is that they are polygynous. So you can have multiple queens in these nests. So if you say there's 20 queens, you've killed 10 of them, there's still 10 they're producing. So you've got to get enough material back into these massive nests to kill every breeder that's in the nest. So that's another trade-off for us.
Okay, thanks.
Yeah, it's fascinating. The other thing I love about this whole thing is we're in a desert. We have citrus from Asia. We have ants from Argentina. We have pests from India and Pakistan. It is a crazy ecosystem that we have built out there. And we irrigate it all with water that we don't have anymore.
I think we have time for probably one
Yeah,
more.
okay,
We're going to get
sure.
you lunch on.
Yeah.
So regarding the hydrogels, because you mentioned that the honeydew product produced by the psyllids sort of can encourage fungal growth on the trees themselves. Is there a risk that the large sucrose solution present in that hydrogels could cause localized fungus where growth where it gets placed in orchards?
Yeah, so all that gel is put on the soil, and the longevity of the gel is 2 to 3 days. If the irrigation comes on, you might get an extra day, half a day, because they will re-swell again, then they go down. So there is, I know, given how xeric it is under those trees when there's no irrigation, I just don't think there's a chance for those molds to develop on anything that's growing in the soil. The reason they do well on the leaves is the honeydew's being produced all the time, especially by the ones that produce the sugar water, like the soft scales and the mealybugs, and it's on the leaf with its own like little microclimate around the leaf, where the humidity is a bit higher as well. Yeah, so I don't think we need to worry about that in the field.
Thank you.
Okay. All right, thank you very much.
One more time.